This is Madam Rose. Today, we are going to see the process of protein synthesis which takes place within our body. And this lesson is prepared specifically for Form 6 students. And before we proceed, let's see the objective of the lesson. By the end of the lesson, students should be able to understand what is amino acid and the protein. Also, the student should be able to explain the difference between RNA and the DNA. And finally, the student should be able to describe mechanism of protein synthesis. Definition of key term. Amino acid is the building block which used in manufacturing the protein. While protein is the large molecules that are formed by combination of several monomers and a condensation process. And protein synthesis, this is the process in which there is several monomers of amino acids join up and a condensation process, hence formation of polypeptide. Ribonucleic acid. This is the kind of nucleic acid formed in the nucleus for the purpose of assisting DNA in the formations of polypeptide. This ribonucleic acid mainly found in the cytoplasm of the cell, and its work is to carry information, to carry amino acid for the purpose of protein synthesis. There is three kinds: transfer RNA, ribosomal RNA, and the messenger RNA. All of these assist DNA during the process of protein synthesis. Messenger RNA, starting with messenger RNA, this carry information from the nucleus to the ribosomes to what exactly kind of protein to be synthesized. Ribosomal RNA, this is assist ribosome, act as a benchmark in the cytoplasm of the cell to assist ribosome to perform its work precisely. While transfer RNA, its work is to transfer amino acid from one place to another for the purpose of formation of polypeptide. Structure of DNA. DNA is the deoxyribonucleic acid. Its work is to determine what kind of amino acid, what kind of protein to be synthesized within the body. Its structure, as you can see, it's helical. It's folded, it's not linear, and it's double-stranded, means it have two strands that run antagonistically. Its compositions, it consists of the phosphate from phosphoric acid. It has a sugar, pentose sugar, and the base. This base is adenine, thymine, cytosine, and guanine. This is nitrogenous bases, and this base used to touch each other by hydrogen bond. When adenine is attached with thymine, there is two hydrogen bonds that bond them. And when thymine and guanine is combined, there is three hydrogen that bond them. This hydrogen is not strong. They are weak. They can be broken by either heat or by mechanical forces. This is how this base is arranged by using base pairing rule. This is an activity for learners. This question will be asking to learners to know if they know this mechanism of protein synthesis before I teach them. So here there is a formation of messenger RNA, formation of peptide, transcription and translation. So I'll asking the student to arrange this process from point 1 to point D. So I hope the student will be giving answer like mine. My answer is, there is two processes of protein synthesis, transcription and translation. Under transcription, there is formation of messenger RNA, and under translation, there is formation of polypeptide. Therefore, when you come back in the previous slide, process number 1 is transcription, so C will be A, and under transcription, there is formation of messenger RNA. So C will be A, and A will be B. Under translation will be formation of polypeptide. So D will be C and B will be D. So this is the process of protein synthesis. Mechanism of protein synthesis. As I said earlier, mechanism of protein synthesis involves two steps: transcription and translation. Under transcription is where you found formation of messenger RNA, and under translation is where polypeptide formed. Therefore, this is the structure of nuclear. This is nuclear pore. Transcription process is taking place within the nuclear, where messenger RNA is formed under transcription process. Therefore, this messenger RNA exits nuclear through nuclear pore, under this nuclear pore, to the cytoplasm of the cell. Within this cytoplasm of the cell is where you can find the ribosome. This is the ribosome, structure of ribosome. When messenger RNA exits the nuclear to the cytoplasm, it finds the ribosome and it lands in the ribosomes. In the ribosome, there is a portion that exists between a large subunit and a small subunit. This is the place where messenger RNA lands. Okay. Therefore, transfer RNA that is located in the cytoplasm, it carries amino acid that is suspended in the cytoplasm and came here in the ribosome to unlock the information that is carried by messenger RNA, in which it determines what kind of protein to be synthesized. So the transfer RNA will be carrying 3 codes, in which it came and read the code that is carried in the messenger RNA to determine what kind of protein to be synthesized at that time. After transfer RNA read this information from messenger RNA, it leave this amino acid to combine with another under condensation process to form polypeptide chain. As you can see, this is the structure of polypeptide chain. And when this amino acid in the cytoplasm reach to the end, meaning that all the amino acid released will be joined up to form a large molecule of protein. And this is how protein is formed under translation process. Question. For more clarification and understanding, you can attempt this question. Thank you.